Record.
Okay.
Did it say recording? Okay, do you wanna?
It's turning on. Or at least it should be turning on. Did it work? Why is it not?
What are you doing?
All right, we're good.
All right, everyone, welcome to the week 1 review session. Happy Valentine's Day. All right, so we're just gonna go ahead, right?
Yeah, I'm just gonna pull up the chat here.
Let us know if you guys have any questions throughout the review session, like at all, since this is like such a small group, like any questions at all, don't worry. Okay. And then at the end of the review session, you guys can ask like individual like poem set questions, course questions, like anything else that's on your mind. Yeah.
All right.
Reminder, you do have your problem sets due tomorrow. So if you do have any last minute questions, John and I can answer those.
Also, don't get in the habit of like expecting to do them on Tuesday night because they're gonna be due on Sundays from now on.
You guys didn't hear anything, right? No. Okay, good. Yo, guys, come on. You guys are totally not with me. Is this a discussion? Oh, hey, what's up, John?
What's up? But yeah, so moving forward, the problem sets are all gonna start being due on Sunday nights at 11:59. Laura has the grace period for emergencies and stuff like that. But really don't get in the habit of, thinking, oh, Tuesday night, we're good. Please don't do that. All right, so we're gonna get started. Topic 1 stuff, talk about protein structure, amino acid structure. Laura went over this for the last couple lectures. I know we didn't start doing content really until like the second lecture or so. But so if you look on the screen over here, you have the amino group on the left side, you have the carboxyl group on the right side, and then on the central carbon or alpha carbon, however you want to call it, there's going to be an R group present. And that R group is what's going to characterize the amino acid depending on its chemical and biochemical structures and relativity. All right, so formation of the peptide bond. We have over here a pretty basic drawing of just 2 random amino acid structures. Is that bond? I forget. Glycine, yes. It's just the H as the R group. So important to note here, it's a dehydration reaction. We're mostly thinking about the protein like strand being formed. Very important to remember that there is a water molecule being formed also when the ribosome does that. Key factor here, this new C to N bond is formed, peptide bond, pretty straightforward. Has different chemical functions and also different chemical reactions when compared to a single carbon to nitrogen bond that you'd see elsewhere. It is shortened. It has basically double bond structure and function, and it is a fairly strong bond comparing to the other bonds that we've been over in class. And the breaking of a peptide bond, aka the reverse of the peptide bond formation, is a hydrolysis reaction. So for the formation of the peptide bond, we were forming water. For the breaking of the peptide bond, you're going to need that OH on one side, that H on the other side. So you get the 2 amino acids split up. Any questions? So far good? Okay. All right, polypeptide anatomy. So to make it easier for not just students and people that work with it to learn it, but for everybody, each amino acid on a polypeptide chain is labeled with a number name. You guys don't need to go over the like the 3-letter, the 1-letter names. You also don't need to know the exact structures of every single amino acid present. It is important for you guys to know the different groups that they are part of, but we will go over that in a couple of slides. So the R groups are arranged by chemistry and size. If you look here on this polypeptide chain, on one side the R group is going to be facing down, on the other side the R group is going to be facing up, and that goes like that concurrently until the end of the peptide chain. That is important for new levels of structure that we will also go over in just a couple of minutes.
Okay, so why are they orientated like opposite to up and down?
It doesn't have to necessarily be 100% up and down. It's just they won't be in the same general area. We will be talking about sterics and strain at some point too. But it's just important to know that they won't be like in the same general area, especially if they are either like hydrophobic amino acids that will be having the
Is
like,
it also like
repelling
when it
forces.
folds, like the ones that are usually sticking up are like hydrophilic, the ones sticking down are hydrophobic?
not necessarily. I
Is
wouldn't
it
think
more
of,
like electronegativity where if they're both pointing in the same direction, they kind of don't like that?
I wouldn't think about it as like a hydrophobic hydrophilic type of thing. Usually what happens is there are more hydrophobic and hydrophilic sections of the polypeptide chains that we go over. This is just a very small like part that was taken out of a larger polypeptide chain. Like Laura mentioned in class, most of your proteins are going to be from the 100 to like a couple thousand amino acid long chains. That's just a basic drawing of it.
I'm just gonna jump in. So basically, in secondary structure, which is when we like sort of look at like the way that the R groups are oriented, you have that, which we'll go into. You have alpha helices and beta pleated sheets, and the R groups are oriented in different ways in both of these structures. In alpha helices, they stick out of the helix, and in beta pleated sheets, they alternate. This does have to do with like size and like steric hindrance, but also like you were saying, like in an alpha helix. Some of the sometimes it's oriented in a way that like half of the helix is more non-polar, half is like polar. Same with the beta pleated sheets, but it's not really something that we really need to go over in class. It's also it's still sort of like the way that the R groups are oriented is still, I guess, like not particularly known. It's also not really known why some alpha helices form and other beta pleated sheets form, or like, yeah. So it's a really good question, but I think all you need to know is just that the R groups are not oriented just like you see in like the shorthand notation. Just know that in like an actual 3D protein, like it's not like all the R groups are facing down, they're like around.
Also important, Laura mentioned this in class, peptide bond, like I said, shortens that last C to the end. And that happens because of the formation of the peptide bond, but also very important aspect of that is the resonance present. Laura kind of drew it on the board in lecture a couple days ago with like the dash lines, or she mentioned it. I don't know if she drew it. But just think about electron movement, electronegativity of oxygen is stronger than carbon, and just think about how the electrons can move on from one of the oxygens to the other to form this kind of dashed drawing that you can have.
So what we do is we just record, upload the recording in the YouTube channel that the TAs have, which I think is in.
Yeah, if it's not Moodle, I'll just send an email out.
Oh,
after.
it is? Okay,
Yeah,
cool.
okay. Yeah, we'll be uploading the review session tonight.
Yeah.
Yeah. All right. So primary structure, very important basis of your protein without it being folded. It is the sequence of amino acids present. we're not really looking into how it's gonna fold, what functions it's gonna form as of yet, just for primary structure. You're just thinking of the order of amino acids, and that the peptide bond holding them together is very strong. A lot of proteins perform like that. Protein denaturing is like very complicated process for the cell to like get used to. So it doesn't happen. Proteins serve their functions according to their structure. So you will you'll hardly see like a case in class that Laura's gonna be like, oh, the protein broke, guys. No, she's not gonna say that. All right. Secondary structure, like Rossella was talking about earlier, we have the formation of these different structures. You have R group interactions here that are the main cause of this type of structure to be formed. We have alpha helices, we have beta pleated sheets. If you look at the ribbon structure that Laura went over in class, the alpha helix looks like the ribbon, which is why the ribbon format's called that. The beta sheets look like they look like a little ladder going down. And like we were talking about on that drawing on the slideshow, the amino acids on the beta pleated sheet have their R groups facing different directions, which is why they have the little stair looking look to it. The alpha helix has the curves being formed. Laura talked about the hydrogen bond formation that leads that to occur. So if you look at the board here, you have the amino acids, R groups forming hydrogen bonds, which is what's holding on to their secondary structure. And so there's different types of bonds that can happen in protein formation. Here we're talking about dipole. So we don't go over the reason why hydrogen bonds are, or how the hydrogen bonds are formed, or like what the necessity is, right? We don't talk about like covalency to end, right, in this class.
Oh, well, it's
That's
first,
fine.
like a non-polar.
Yeah,
A little bit of
they need to be polar for the hydrogen bonds to form, because they are a polar bond, or they are a polar interaction bond being formed, and they are non-covalent. Remember that. Non-covalent bonds form that sort of interaction. But yeah, secondary structure, alpha helix, beta sheets. We will talk about in class why those are important and how that sort of protein formation is important to some function of itself. Yes.
Exactly. So secondary structure only involves the backbone. There are no R group interactions when we talk about secondary structure. Can I just jump in for a second?
Absolutely.
So in secondary structure, it's really important to know which atoms on the backbone are involved in the hydrogen bonds. So remember, in hydrogen bonds, you need a hydrogen that has like a partial positive dipole, which can only happen when the hydrogen is bonded to an electronegative atom, such as nitrogen or oxygen. So let's go back to that first slide. So this is over here, this is the backbone of the amino acid, and this is the variable R group. So in secondary structure, we're only looking at this backbone. The hydrogen bonds that occur are between amino acids that are not adjacent to each other. So it would be like this amino acid over here and another amino acid like below it in the helix or in the beta pleated sheet. The atoms that are involved in hydrogen bonding, it's important to note, it's this hydrogen over here, or the atoms that are involved are this hydrogen, these oxygen atoms, and like this nitrogen one. So this hydrogen can hydrogen bond with like this oxygen from another amino acid, or like this nitrogen can hydrogen bond with a hydrogen on another amino acid. It's important to note that this hydrogen over here bonded to the central carbon does not have a partial positive dipole because their electronegativities are similar. So this atom is not participating in secondary structure hydrogen bond formation. So it's only the amino groups and the carboxyl group amino acids that are involved in the secondary structure. So if you get a question that says like which atoms are involved in secondary structure, it's important to say not this one because the hydrogen bonded to the central carbon does not have that partial positive dipole. Does that make sense? Okay. Tertiary structure.
Tertiary structure, guys. So in a couple classes, you'll hear Laura talking about subunit. That's just basically a name for the polypeptide chain being folded. So it's the overall 3D shape of a single polypeptide chain, and polypeptide chain protein. So the interactions are based on R group chemistry. Like Rossella was talking about, the backbone atoms forming the hydrogen bonds. It's important to note that for this specific level of structure, it's tertiary structure only. We're going to be talking about salt bridges or disulfide bonds being formed by two cysteines. You don't need to know cysteine structure. You just need to know that cysteine does that. So if you look at this drawing right here, you have disulfide linkage. You have a hydrogen bond being formed. This time, not by the backbone atoms, but by the R group. And then you can also have ionic bond formation that, like the slide was talking about here, there's some sort of hydrophobic interaction going on in there also. So it's overall how the protein is being shaped in a three-dimensional way. And we were talking about polypeptide chains on primary structure. That's non-folded. This is folded. Laura is a big believer in, not just Laura, it's like a major thing in biology, but protein function is done because of structure. So if there is a change in structure, there will be a change in function, which is why denaturing is a bad thing, because you're expecting protein to be in its normal folded shape, and once it's not, it cannot perform its function. So again, just take a look. This is the backbone, and overall, you're thinking about. Laura likes for you to think about protein structures in your head, like a three-dimensional way. I don't know, it's kind of like hard to think about in the like when you first start doing it, but as you get into like more, as you understand it more, it gets easier to do. Oh, they are very common. They're like a, if you look at like the way a protein folds three-dimensionally, R group interactions are what lead the protein to fold that way. Also, there are chaperone proteins involved in protein folding, but right here we're just talking about specific cases of R group interactions that will lead to protein structure. Yes.
So yeah, so tertiary structure is only looking at the R group interactions, right? So you have primary structure, it's only between adjacent amino acids, like it's just looking at what's in the polypeptide. Secondary structure is backbone. Tertiary structure is finally like the R groups are interacting, which is what gives it the 3D shape. Yeah. Do you guys have any questions about like the different types of bonds, van der Waals, ionic, hydrogen bonds, sulfide bonds? Any more questions?
So polar charged R groups interactions, they form ionic, right?
Yes.
And then polar uncharged, they form van der Waals, right?
Polar uncharged are hydrogen bonds. And then the non-polar ones form the van der Waals.
Oh, non-polar.
Yes. And then the disulfide bonds are only formed between cysteine amino acids, which we have a slide on that also, I believe. And though that it's important to note that disulfide bonds are found like normally in the center of the protein, and this is because first of all, the bond itself is nonpolar, but also because it is such a strong bond that when you think about it, it makes sense that it would be found in the center because it's like holding the structure together. Something that's interesting, well, because it's also a covalent bond, it's not non-covalent, it's like a lot stronger, so it's more resistant to temperature changes,
pH changes too.
pH changes, all those changes, it's more resistant because it's so strong. So when you see, like, there are a lot of these things called thermophiles, which are organisms that need to exist in like these really extreme, like hot environments. And you'll see that they have a ton of disulfide bonds in their proteins because they keep that structure more, because it can't really denature as much, because denaturization does not destroy like the sulfide bonds. That's a little thing about the sulfide bonds. The hydrophobic effect. The hydrophobic effect is not a bond. It's just basically explaining why a protein folds in a way that the hydrophobic amino acids are in the center and the hydrophilic amino acids are on the exterior. So hydrophobic effect is not a bond itself. Like it's not a bond itself. It's just showing or explaining why the protein folds the way it does. Does that make sense? So the hydrophobic amino acids want to be in the interior of the protein because they can't interact with the hydrophilic aqueous environment in the cell. All right. Any questions?
All right, quaternary structure, really important. Tertiary, quaternary, not the same thing. I know you'll read more into like your topic notes and you'll read into like literature sometimes and you'll think, oh, sounds like the same thing. Not the same thing. Quaternary structure needs multiple 3D formed proteins to come together to be formed. So a single protein, like one strand of amino acids, will never be quaternary structure. It will need another at least another one string of protein to be formed together. Here we have hemoglobin. It's a great example of that. There are four domains. There's two different subunits. There's the alpha and the beta. And it's exemplified in two different colors for you to be able to see that. So back to the text, when multiple polypeptide chains that have folded into tertiary subunits interact together is when you get quaternary structure. Don't be tempted by saying that they're the same thing as one is just like more or like larger. They are like different independently folded proteins coming together into they dimerize, right? That's the term for it.
Dimerizing is like liquid dark.
Forget about dimerization. And so it's two different independently folded proteins coming together. And so not all proteins have quaternary structure. If it's just one protein, it's going to be tertiary. And that's important to know. You probably will get some sort of question on some problem set or some exam asking you about quaternary structure and that being present. I think hemoglobin is a great example if you want to look more into it. Insulin also has different subunits. So if you want to take a peek later on, you can. This interaction also occurs via R group interactions, via the different tertiary structure amino acids that are present. Like Rossella was talking about, when the protein folds, you're typically going to have an interior that's more hydrophobic. You don't want those amino acids coming in contact with aqueous solution. It's no bueno. They tend to stick together after that too, but they, you know, protein misfolding is bad. And then that is why it is tempting to think that they are the same thing. They have the same bond types as tertiary structure, but please somehow get it in there for you that you need multiple tertiary structures to form a quaternary. Do you want to add something?
You guys have any questions?
Yeah, we're gonna have a slide on
Oh,
that,
yeah,
but if
that's
you
like the
want,
next
yeah,
one. Okay. Well, just because that one, that's basically what you just said.
oh, let's get that. Okay,
Did you wait? The bonds that you see in tertiary structure, so van der Waals, hydrogen bonds, disulfide, ionic bonds, those are the same bonds that you'll see in quaternary structure. So the polypeptides that are held in quaternary structure are being held together with the same bonds found in tertiary structure. All right, yeah. You can go to the domains and subunits.
okay, domains versus subunits. I like this topic, because I have to correct the question on the one exam at one point. So subunits is a subunit is basically another word for a folded protein that is a single polypeptide chain. So here you have like a strand, and there's like one single polypeptide chain. It is divided into two different colors, and these are the two different. Is that like shadow, or is it like a third different part?
Where?
On the green, looks purple. I'm assuming it's just shadow. Anyways, so if you look at the left side, you're gonna have a single polypeptide chain that is gonna be colored in two different ways. All that is showing you is that that subunit has two different domains. By the definition, you can say that a domain is an independently folded part of a subunit. And basically what that means is a single part of that polypeptide chain is gonna fold, and then another part is gonna fold on its own. And they're gonna be part of the same chain, they're just gonna be altering functions, and they're gonna have different folding. And then again, subunit, knock it in there. It is a single polypeptide chain. Domains are present in subunits.
You still have questions?
Got it? Good. Good. Okay. Is that good? Yeah. Independently parted or independently folded parts. Remember that. R group interactions, bonds, more amino acids, etc. Oh crap. All right. Types of R group interactions. Beautiful amino acid chart for you guys. You don't need to memorize every single one unless you want to go to med school, then you'd like to do. But that's a story for another time. So you have different categories for the different amino acids that are present. So there are the 10 non-polar amino acids on your top left. That happens because they have non-covalent bonds or covalent bonds formed within each other, and then they end up being hydrophobic. They don't have any polar side to them, which is why they're called non-polar. They will form van der Waals interactions. They will tend to stick together in aqueous solutions. They do not like being near water since they're hydrophobic. And that also occurs because the electronegativity of these different R groups is roughly similar. And so they attract to each other and they are repelled. Polar charged R groups, those are the ones on the right side. You can have a positively polar or a negatively polar charge. You don't need to know every single one, but if you see acid at the end of the name of the amino acid, it's like negatively charged polar amino acid. It's good rule. Yeah.
Van der Waals interactions are considered covalent, right?
No, they're not.
No.
Non-covalent.
Well, they're interactions that occur between non-covalent or non-polar amino acids. They'll be like standing together, not standing, but floating around together, and they'll just be interacting with each other. There'll be repulsion forces too, but I don't think we talked about those yet. But yeah, again, back to the negatively charged. If you see negative, or I mean, if you see the word acid, aspartic acid, glutamic acid, it is a negatively charged amino acid. Also, you might hear the words glutamic acid goes by glutamate and aspartic acid goes by aspartate. So if you hear that, that's what it's referring to, the acids. And then you have the positively charged amino acids, lysine, arginine, histidine. You don't need to know their structures. Why is it important that these are labeled separately from the nonpolar and the polar parts of the chart? They will typically make ionic bonds because of those negative and positive charges. Like that lysine will have that positive sign on the amino group. Aspartic acid will have that negative sign on the oxygen, and they will attract together. And this will form electrostatic interactions between the R groups. Polar uncharged R groups, that blue section that you see right there, some of them are special and like some different pHs that we're not going to mention. They differ from their positively charged and negatively charged because they don't have an actual charge on them. But they are able to make interactions in aqueous solutions. They are not hydrophobic. So if you look at their structure, you'll see oxygens, you'll see nitrogens, and that is why they're able to make these polar interactions. The slide says lots of carbon oxygen, lots of carbon nitrogen, oxygen nitrogen, or I mean oxygen hydrogen, nitrogen hydrogen. There is a large difference in electronegativity depending on the R group that you have. And then there's also size differences. Like a serine is a much smaller amino acid than like a tyrosine, and you have that phenyl group in there. Some of the bulkier amino acids we will talk about in sterics and strain later on. But anything you want to add?
Do you guys have any questions?
Which ones available? Is it a polar attraction?
Yeah, so I think it would be easier to like draw something. But basically what we're looking at here are the different types of R group interactions. So you have two different types of interactions. You have covalent and non-covalent. Covalent means that you're sharing electrons. So if you see over here, these lines in between all the atoms mean that it's a covalent bond. They're sharing those electrons and they're tightly bound together. The only covalent bonds that you see between R group interactions are those disulfide bonds, which only occur between two cysteine amino acids, right? So what happens is that a sulfur, well, the two sulfurs from the two cysteine amino acids will create that covalent bond, which means that they are sharing their electrons. Then you have the non-covalent bonds, which are the rest of the bonds that we talked about. Non-covalent bonds are basically the electrons are being like, they're being shared, but they're not like no, it's fine. This is like when this is a covalent bond, like their electrons are literally being shared in their like electron shells or whatever. But then a non-covalent bond would mean that they have their own electrons, but they're like interacting sort of like they're not like fully bound together, and which is why that they're less they're not as strong. That's not the best like chemistry thing, but that's enough for like the biology.
Yeah. Like maybe assume that most bonds you're gonna see, and it's like, oh, this cysteine is gonna be non-covalent, but it's not peptide either.
Exactly. Right. Yeah. So you only have the only covalent bond that you're gonna see between R groups is that disulfide bond. The hydrogen bonds, the van der Waals, and the ionic bonds are all non-covalent.
The peptide bond is mostly
is covalent, right? Does anybody have any questions just on like what polar versus non-polar bonds mean and like what the van der Waals is? Or is everybody okay with that? Yes.
I see like something on the lines, kind of just like well, I see orange on the line part, like what's all that about.
So sorry, what are you asking
Like,
exactly?
am I like, wait, see, so it's on the left side.
Oh no, this is a polar charge. Polar. So you're not gonna. So over here, these are just the non-polar, which means that they're the bonds in their structure. What is happening is that the atoms have the same electronegativity, which is the likelihood that they're gonna attract the electrons. It's all equal. So all the electrons are shared evenly throughout their R group structure. But what's happening in these ones is that because you have these more electronegative atoms, those more electronegative atoms like oxygen and nitrogen are pulling electrons towards them, creating that sort of dipole. And then over here, you have like the same story, except like more extreme extent where you have like full charges. Like this each has a full charge and can create that ionic bond with like this negative oxygen. So this fully negative oxygen can create an ionic bond with this fully positive hydrogen.
Oh, okay, this try polar.
Yes.
Okay, with diagram was said that don't go like non-polar child.
Oh no, yeah, no, they're. Oh, I see what you're saying. No, like they're just like separated. Like the
yellow's
these don't have,
exclusively
like-
non-polar.
Yeah, sorry. It doesn't mean like polar negative or non-polar positive. It's like they're their own separate groups. Okay.
If they're charged, they're polar. Yeah, I see what he means now.
Yes.
So, the R groups, like, they can form the hydrogen bonds, and then the backbones can also form the hydrogen bonds. So
Exactly.
the backbone interacts with the R.
No, so the backbone only interacts with other backbone atoms. So yeah, it is a little confusing that the hydrogen bond is involved in both structures, but it's just important to know that in secondary structure, hydrogen bonds are occurring in the backbone part and like with the amine group and the carboxyl group. And then in the tertiary structure, the hydrogen bonds are occurring only within these R group structures between like polar, like this oxygen can hydrogen bond with that hydrogen. So yeah, so the hydrogen bonds are very important because they occur in multiple structures. I don't think we need to go over this, but this is just like disulfide bonds again. They're covalent, only occur between two cysteine molecules, and they are found in both tertiary and quaternary structure. And it is the only covalent bond in tertiary and quaternary structure. So in quaternary structure, if you see disulfide bonds in quaternary structure, which means like multiple subunits together, it means that they're most likely like permanently bonded together, the different subunits, because it is so strong. So if you see like if there are two like polypeptides that need to like sometimes bind together and sometimes like dissociate, you probably won't see many disulfide bonds in that quaternary structure. Okay. This is just for you guys for later on if you like go through the video again. It's like a summary slide of the different structures. It says like the structure, the description, and then what bonds are in those structures. And it divides them up between the covalent and non-covalent. Okay. This, I love this diagram, this one over here. It is so helpful. So basically in this course, you're not supposed to memorize all of the amino acids. You're not going to be given like an amino acid and you're not going to be asked like, what is this? Like,
draw it.
But yeah, but what you are going to be asked to do is to understand the chemistry of the amino acids and be able to explain why alanine is nonpolar, why threonine is polar, and why glutamate is charged. So you don't have to explain like the structure, like you don't have to explain or draw with the structures, but you have to explain why it's labeled as polar, nonpolar, charged. And the way you do this is by like talking about what types of bonds are there. Do you see nonpolar or polar bonds? Like what sort of atoms are there? The size.
The electronegativity present on the molecule.
Exactly. This graph is really helpful. You get a cheat sheet for your exams. I would put this on there. I would like take a picture of it right now. I love it because basically, when you see these charts that like divided between nonpolar, polar, electrically charged, all of these amino acids aren't equally polar. Some are more polar than others. And the same thing with these nonpolar ones. For example, this amino acid is more nonpolar than glycine. And the reason why is because the more nonpolar bonds you have in an R group, the more nonpolar it is. And over here in this chart, you see it's giving you this like hydrophobicity scale, which, so when you're told like, okay, if you have I can never say it, you have phenylalanine and alanine, which one's more likely found in the interior of the protein? The way you describe it in your answer will be like, well, phenylalanine has more nonpolar bonds than alanine, so it is more nonpolar, so it will be more likely found in the interior. It doesn't want to be like closer to the hydrophilic water, whatever. But just to second, this is not like on your exam, you're not going to say like, oh, I use this. That's how I know, but it's a good check to see like, okay, yes, where's phenylalanine?
Top fourth.
Phenylalanine is more hydrophobic than alanine, and it says it like right there. So it's like when you're looking at these amino acids, you have to more think like of like a whole like what do they call it? Like
Spectrum.
a spectrum of hydrophobicity. Okay, and yeah, so this will help you like in your questions, like which one's more likely found in interior, exterior. Just to like second check to see if you got it right, but it's not like you can't like say your reasoning because of that. But yeah.
There we are.
Yes, sorry.
So you're saying that we're going to ask why amino acids are blended hybrids, right?
Mhm.
So in terms of acidic with the charges, how do we spend this thing that's acidic because there's resonance?
Yeah, so for these types of questions, most likely you will be asked which one's more found in the interior or the exterior. So it'll be more like a question like tyrosine and valine, which one is likely more found in the interior? And then you're gonna say valine because it has nonpolar bonds, and tyrosine has that polar oxygen hydrogen bond. So you just need to be able to look at the structures and determine, like based off the chemistry of their R groups, where they're going to be in the protein. If that makes sense.
Somebody else had a question, right? Or am I going crazy?
Okay, steric hindrance. I think we already talked about this. It's just basically like how the size of an R group can affect the structure of a protein. So if you had a mutation that substituted this amino acid glycine that has a very small R group with this huge one arginine that has a very large R group, there is going to be steric hindrance because it'll change. It'll like that big change in size can affect this the shape of the protein.
Cyclic rings are very bulky. There are
Yes.
a couple R groups that have cyclic rings, tryptophan, phenylalanine. There's like another one. There's tyrosine. They're all like massive. If you look at like how the chemical structure is drawn here, that ring right there is equivalent to like that big carbon that's previous to it. But there's 6 of them. So just think about it, and like if you look at the drawing that you have on that chart, like the previous chart, the green, the gold, yellow, they don't look like relatively big, like when you look at them. But if you take into account the number of carbons present on each of those cyclic rings, you get some sort of understanding that they're massive compared to glycine, which is literally a hydrogen. So just be mindful of that. There's a couple others. Proline is also a big example. The R group ends up bonding to the backbone structure. It's like the only case that happens, and then you'll see it drawn with like the R group being bound to the nitrogen on the left side. And that will come back in the near future with secondary structure information, but we will not be, actually, never mind, it is there, it is present. So if you look at like a 3D folate protein, it has a ribbon, and then it has like a little like side chain to it that isn't like a ribbon. It's most likely because there is a proline present there, and they're called helix breakers. I don't know if you heard that before. I was taught that. And they break the helix structure, and then in some proteins, they are also forming a new helix right after. And that structure differentiation is what's going to cause the difference in function of the protein. So yeah. You have the chart again.
All right, some practice problem questions. And then we can move on to, oh wait,
That's the last one.
oh, it's not, we have this one. Oh, there was. Okay, so practice problem.
Models before?
Okay, fine.
Yeah.
Gotta do them.
Yeah. Oh, jeez. All right. So Laura went over this last lecture. If you were chit-chatting, you probably didn't hear it. No, I'm kidding. There's a couple of them: ball and stick, space fill, ribbon, backbone. My favorite, ribbon. It's the only one that matters. If you think otherwise, well, I don't know. But ball and stick, it's only showing the atom as like a big like circle on the screen with a bond being the stick, which is why it's called the ball and stick model. And it shows the atoms in the protein, including the backbone and the R group. You might be asked a question in the exam like, oh, which type of protein modeling would I use if I'm trying to exemplify why the alpha helix structure is important? And then you'll say, I'm using the ribbon model because it's the best. Sorry, I'm biased. But yeah, we have space fill. Space fill is good if you're trying to like take a look into different proteins that are well known. So like you'll sometimes see like a hemoglobin space fill model, and it will show the hemoglobin and the space where the oxygen is being transported. And it shows every atom, and it's very compressed. It doesn't show how the bonds are formed. It's just like a whole bunch of just atoms being thrown together. Ribbon, best model. It has the ribbons, it has the beta sheets, it shows everything you need to see. It's prettier. But you can't see the R groups. What matters really is the structure in that case, and it's better. Backbone, it only shows the polypeptide backbone. It's like very thin. It kind of looks like wireframe, but it's a little different. It doesn't show the R groups, and it's just basically the outline of the protein if you take a peek at it. And you can also have some models being overlapped. So if you use like space fill, but you want to see the alpha helix structures, you use space fill with the ribbon, and it'll make it 10 times better. But yeah, and that was the last one, right?
Yeah.
And then
Just show it.
those are the pictures. If you look at this and you don't think ribbon is superior, lost cause. All right. Oh, we're going back to the questions.
Okay, so some practice problems. Number 1, which is more likely to be found in the interior of the protein, phenylalanine or tyrosine? I think I already gave that example, but what do you guys think? These are the things. Where is it? Phenylalanine or tyrosine? Which one's more likely to be found in the interior? You can just like shout it out, raise your hand. Yes, exactly. Phenylalanine is more likely found in the interior, and that's because it has that like really large benzene ring, which is full of hydrogen carbon bonds, which are nonpolar. Tyrosine does have that large benzene ring, but it has that OH group, which is polar at the end, which then makes it more polar, so more likely found on the exterior. Yeah, nice. Okay, if a protein denatures, what levels of structure is or is slash are broken and why? Secondary. So just think, when a protein denatures, what types of bonds are broken? Covalent or non-covalent? Covalent or non-covalent? Non-covalent, right? Because those are weaker. So the non-covalent bonds that are broken are hydrogen bonds, the van der Waals, the ionic bonds, right? So if those are all broken, what levels of structure are going to be broken? Yeah, tertiary, quaternary, and secondary, right? Because secondary structure is held together by non-covalent bonds. The only structure that remains then is primary structure, right? Because primary structure is held together by those covalent peptide bonds. Okay, any questions? Question 3. If a mutation occurs that changes alanine to valine, alanine to valine, what levels of structure are affected? Alanine to valine are most likely affected. Yes, primary. Do you want to explain why? Exactly. Exactly. So why wouldn't it affect the secondary or the tertiary structure? Exactly. Yeah. So it's not going to affect secondary structure because the backbones are the same, right? Because every amino acid has the same backbones. It's not going to affect tertiary structure because it has very similar bonds and their sizes are also very similar. So the chemistry isn't changing that much at all. So there was most likely not going to be any effect to the protein folding. But how about if we change alanine to asparagine? Where is that? Alanine to asparagine. What's going to happen? Is the protein going to be, or are the levels of structure going to be affected when it folds? What was that? Alanine to asparagine. Yeah. So because our groups are different, they make different bonds. They're probably going to be found in different areas of the folded protein. So it's going to affect the 3D folded structure. So like tertiary, and it could affect secondary structure too because of that steric hindrance, since this is a bigger R group than alanine. So yeah. All right.
Real world example, guys. So it's asking example. I don't know if Laura went over this, she might have, but just think about how nonpolar interactions occur and how hydrophobic molecules would be interacting with hydrophilic molecules.
Cooking an egg.
Oh, cooking an egg is a good example.
It wasn't the one I was thinking of.
Yeah, well, cooking it. Well, it's not a good example of this. I think what we were talking about. Do you want to just? Yeah. So what the cooking an egg is a good example of protein aggregation when you denature protein. So like when you're cooking an egg, there are lots of proteins in there. So when you heat it up, the proteins are denaturing. Their hydrophobic cores are exposed, which cause them to all like clump together, so that the hydrophobic parts of the unfolded proteins aren't like facing the aqueous environment, so they all clump, and then that's what happens with the egg. But in this one, yeah, the example
I was thinking of, when you buy an Italian stuff like seasoning for your salad at the grocery store, and it's all like separated in layers, it's the oil not trying to mix with the water. when you were young, also, you probably played with water and oil before, like put in the cup, they divide into layers. Same thing. The hydrophobic molecules clump up because they don't want to interact with the water, and therefore the two different layers are formed. That's probably key. Also, detergent. Laundry detergent is probably something that, not laundry, dishwashing detergent is something that we're going to talk about in class. It's useful for dishwashing because not only can some parts of the detergent interact with the water, some parts can also interact with the hydrophobic parts of the products that are left over in the plates that you can wash. Good to think, you want to understand the theory behind it. Sometimes it's also easier to apply it to something you see every day.
Okay, so that's all we had for the slides. I'm gonna stop the. Wait.